David used to be in Bell Labs and then later has been at UCSD for a very long time. So what I asked David to play the contrarian role. So this is the first, and it's a challenge talk. So the idea is that he's going to talk about the vibrissae and somatic sensation, and he's going to challenge us to rethink what could we do about the sort of like findings that he has, and I've seen his talk, but I don't want to spoil it, so I let him to do that, and also to challenge us to see how do we use and utilize the modern tools, for example, in machine learning, to see can we verify those findings, can we extend it, and so on and so forth. So I won't take his time. Thank you, David.
Okay, thanks, Nima. Thanks for the invitation. All right, so this will be mean field versus machine learning, and we'll see how everybody feels at the end. So there is something about open data, I think, that needs to be said that wasn't said so far. The one thing that's great about this is it used to be you'd open up a paper and read it, and you'd see typical best example, and then you'd read the text, and they say, yes, we have n equal 20 other examples, but you can't do that anymore. You have to show the other 20 examples, and you're sort of naked before the world to see your data. And in a serious way, I think this is maybe the most important thing to keeping the field clean. Okay, so I'm going to focus on just one issue. It's an issue that was raised nearly 400 years ago by Descartes in his Methods chapter 5, and it really has to do with the issue of actively sensing an object. In some ways, all of sensation is active. We move our eyes, we caress with our hands, we move our head, we perk up our ears, and if you're a rodent, you whisk. And the example that Descartes showed was a blind man trying to detect where an object is and tapping it at a distance. So not only is it active sensing, but it's trying to detect an object at a distance. Okay, so I think anybody who runs a lab knows you spend your time writing grants, which may not be a futile exercise these days, and teaching classes and answering email. But I'll describe all the published data to date and who did what, but I want to introduce some new data, unpublished data, which I think poses some challenges by Pantan Yao, Ray Liu, and Arash Farisi, who's here. And since part of open data has to do with collaborative efforts, I think it's important to make it clear that much of our work over the last few years has been part of these large collaborations. One is the C19, and I'll show data that comes from Martin Deschênes and David Golomb and Fan Wang, but you should know that Jay Aram and Carell and others at the Allen have been involved in these collaborations. And like I said, these are funded through collaborative efforts. Okay, so let's do this. All right. This is a Canadian mouse. It's friendly. Tim, take note. What's going on? What do you guys see? What's the first thing you see? You're in the lab. You have to plan your experiments. Well, what's special about the way this animal is searching? Moving its whiskers. Okay, what else? What else is it moving? It's moving its nose, moving its head. And what about moving the whiskers? Was there anything special about the motion? And what? Rhythmic. All right, what else? Okay, so this is the point. The first thing we do, right? The first challenge to the experimentalist is you just look, and you have to have some mindset, some approximation that's going to guide your future experiments. So here, the animal is searching. It's searching in the dark. Obviously, this was an infrared videograph. And the head, the vibrissatouch, they're moving. And as somebody pointed out, there's a rhythmic component. So if we hear the word rhythm, right, we want to think in the frequency domain. So we have a signal that perhaps is sharp in the frequency domain in this cartoon. It's what I called rhythm. And then the head is moving, and sometimes there's fast feedback on the whiskers, and sometimes they're their set point is shifting and their amplitude is shifting, and this is some broadband signal. So superficially, it seems like there's two components of the movement. Now, in principle, this could be one motor control signal, or maybe nature chose to split this up as the sum of multiple signals, perhaps even a rhythmic signal. So let's just go back in time. This is thesis work of Jeff Moore, who's now a prof at USC, and understand how we were able to determine the circuitry for this behavior. So on the top line, okay, what we see is that there's a period the animal is breathing, and that's shown in red, the oscillation in red, and it's whisking, or at first it's not even whisking, in blue, and then it starts to sniff and whisk at the same time. And it seems to be correlated. This was discovered by Wally Welker, 1964, a while ago, okay? Now, the question is, this could just be one oscillator and a switch, okay? We want to see if we could break it. So if you put ammonia on the nose, okay, it'll actually will end up shutting down the breathing complex. It's called the pre-Botzinger complex. The mechanism was something we didn't figure out to Amalia Collado Perez. Figured this out only a few years ago, but we shut down the breathing complex. The animal is still whisking. So that tells us that there is a whisking oscillator which is separate from a breathing oscillator, okay? So I think, as everybody knows, you milk behavior as much as you can, okay? The next thing is something that Jeff found, which is that there are epochs in time where the animal will whisk quickly and it'll breathe slowly, and that's shown on the bottom line. So the blue, again, is whisking, and it's kind of winding down in amplitude, and then, boom, you breathe and you reset the whisk. And you breathe and you reset the whisk. Okay, this is one example. There's about 18,000 examples in the paper. It's long published. And to go on, okay, so we know something about the order, that there's a separate oscillator. So now, how do we discover things about the oscillator circuit itself? Okay, so this is intracellular data, okay, that was taken, actually this was taken by Martin, and we were able to activate this oscillator in a slightly anesthetized animal, take that as you will, okay? And if you patch onto these cells, dirty patch, what you find is that there are some units that are in phase with whisking. They're spiking as the whiskers protract. There are some neurons that are spiking as the neurons retract, okay? In both cases, every cell, when we chase it with in situ hybridization, is an inhibitory cell, okay? So Tim Harris, our good friend, of course, came up with neuropixels. This is not a neuropixels experiment, okay? This is a neuropixel experiment, because you need to record from one cell, and then you need to get back to that cell. You need to fill it with a dye, and you need to find out if it's an inhibitory and excitatory cell. So we label one synapse. Okay, just to give you a flavor, okay, at this point we're kind of stuck, and we need to understand something about the sign of the connection strengths from the breathing oscillator to the whisking oscillator. Classic physiology has run out. This is work by Jun Takano, Vincent Provasto, who's here. Fan Wang, this is data taken in Fan Wang's lab, made an animal in which the enzyme Cre is split into two pieces. One is intrinsic to inhibitory cells in this animal, parvohuman cells. The other we inject into the facial motor nucleus with a lentivirus that goes retrograde, as shown here in the little box with the green dots in it. Okay, we could label cells within this vibrissa oscillator. But then more importantly, once we convince ourselves that the right cells were labeled, okay, we could also use Cre that's expressed in this vibrissa oscillator to express a protein, a receptor that then pseudotype rabies will bind to, enter the cell, go backwards into the breathing oscillator, go backwards, and we could label those cells and again do in situ hybridization. And we find out this system works with a breathing oscillator inhibiting a whisking oscillator that then could inhibit the facial muscle. So it's an inhibition of inhibition. That's two negatives. It gives you a positive sign, and therefore whisking and breathing when the animal is excited are in phase. Okay, a fair amount of work stretched over a decade, but we end up with the following picture. So again, remember what the behavior is. You could get a reset due to breathing, that's shown in red, and then the oscillator just sort of winds down but keeps going for many cycles. You have two populations of inhibitory cells, I called retraction, protraction, that make connections among themselves and between themselves. And then there's an input that comes down from the pre-Botzinger complex. And because it's inhibition, okay, you need to have a positive signal, an excitatory signal, to thrust the vibrissa forward, because, I mean, they're basically doing the butterfly stroke, okay? And then they project down to the facial nucleus. Okay, so this is great, but we want to, instead of just being an amalgam of experimental facts, we want to turn this somehow into something simple. We want to get a principle out of it. So how do we model this? Okay, how do we model it? Well, one possibility is brute force, Hodgkin-Huxley. Write down conductance-based equations. In some cases, we know the details. In other cases, we make our best approximation to the detail. This is called posterior probabilities. We just made the best approximations. At the end of the day, we put everything in. This is the work of David Golomb. It's part of our long-term collaboration. And what you see is we could come in with a breathing pulse on top where it's labeled pre-Botzinger. And in gory detail, we can predict the behavior that we see experimentally. Now the question is, what have we learned from all of this? Is there any principles? So I would say that by putting this together, there's some kind of self-consistency argument. This gets back to work by Peter Getting a long time ago in central pattern generators, in invertebrates. You put everything together and you see if the parameters kind of make sense. So you probably didn't leave anything out. But we really didn't gain anything that gives us some idea of how nature likes to build circuits, okay? And one thing that you could do is sometimes you have enough details about the system that you can start to average over the system. This is the beauty of mean field theory in physics. We know a lot about a spin system, okay, but we don't want to describe all the spins. We want to average, okay, over as many quantities as we can and get a formula that tells us something we can measure in the laboratory. So this is what we did, and this will lead to a challenge. We're going to look at one particular case here, which is the case of a free-running oscillator. We have these two populations of inhibitory cells, the ones I called protraction, retraction, and we're down to a very few parameters, okay? The interactions, okay, are the interactions between these populations, inter, and the interactions within the population, intra. There's an external input that comes in, okay, that's 3 parameters, and then there's a slow current, this adaptation current. That's 4 parameters. Okay, that's it, all right? What we derive are formulas, and what I've plotted here in green are the results of those formulas, rather than put something ugly up here. And the beauty of formulas is they tell you what quantities you depend on, and they tell you what quantities you don't depend on, okay? So what we find, for example, is that we could derive the phase plane for this oscillator. So there's a region where the connections are weak, and there's a single relative strength, which is the connection between populations, inter minus the connection strength within the population. And as a function of that parameter, when it's very weak, the neurons just do their own thing. It's uniform. When the connections are very, very strong, the system is bistable. One group fires or the other group fires. And in the middle, that gray band is a region where it could oscillate. Okay? So the bistable region is like binocular disparity, where neurons fire or don't fire. But in this middle region, as a function of this interaction strength, we could vary the frequency of firing, but the amplitude is unaffected. Okay? On the other hand, if we go to this oscillatory region shown on the right, what we see here is that as we vary the external input, the frequency stays constant, but the amplitude of whisking changes. So what we've learned from this mean field approach by averaging is that nature is modular. There's one parameter that controls frequency. There's another parameter that controls amplitude. They're not mixed. So the lesson here is that it seems like it's a very general circuit, but it has a very particular phase, which is oscillatory. As I mentioned, we've shown that frequency and amplitude are independent. Nature is modular. Now, the challenge here is, the formulas and this phase diagram, is that strictly needed to gain insight to the circuit? Okay, obviously, we felt it is, but you may have a different opinion. And what I want to know is, if you didn't take a class in stat mech, and so you weren't thinking about mean field theory, okay, could you elucidate this kind of modularity out of a machine learning approach as opposed to this more traditional mathematical approach that we use? Okay, challenge one. Okay, I want to move on to this next topic. It's unpublished work, and I want to talk about as the animal is touching something, like I showed you with Descartes' blind man with the stick, can the animal actually learn the coordinates, right, in two-dimensional space? It could be x, y space. Over here, I'm going to use sort of polar coordinates, angle, and where you are, phase in the whisk cycle. This is a topic that's been addressed for a long time, starting with work from the late John Curtis. You notice about a quarter of these papers are from Carell's group. But I really want to focus on this one particular question: how does the rat or how does the mouse know where the object is in space? So again, let's go back to behavior and let's make sure that a couple of things, simple things, make sense. Okay, so the rodent is going to be whisking against a pole. The pole is moving all over the place. We could sample all positions along the length and all phases within the whisk cycle. And the important point here is that the animal just whisks through, okay, until it slips off. So what that means, if it contacts the pole early on, it's going to stay in contact with the pole rather than if it touches it when it's almost all the way protracted. And consistent with this, experimentally, we find that the contact duration, of course, is shortest phase at initial contact. If that's 0 at the very beginning, it's the longest contact. If it happens at pi all the way forward, it's the shortest contact. So naive ideas make sense in terms of data. And critically, we want to measure the torque, okay, because that's what the animal knows from touching. And just for reference signal, it's coming in at around 10 to the minus 7 Newton meters. Okay, so experimentally, you can only measure a restricted number of parameters about behavior. We saw this nice talk on that earlier. So we also did detailed modeling. Again, this is work with David Golomb and work by Ray Wu in my lab and Arash Farizi. The critical thing is we can make a mix experiment calculated model. So the torque as a function of where you are on the whisker, okay, falls off along the shaft of the whisker. This seems backwards from a stickball bat, but the whisker is tapered, okay? And it falls off roughly logarithmically. Okay, now the torque also depends on where the vibrissa first contacted the pole. So the torque, if it contracts near the protracted end, the torque is very small. If it protracted, all of a sudden all the way back, it retracted, the torque is very high. We could flip this, and we could also make plots as a function of the initial phase of contact at different distances along the shaft, and this sort of falls off sort of like a cosine curve. Okay, so we basically have a picture of torque as a function of these polar coordinates. Now, the problem is, okay, so we've learned that torque is some function of exponential distance and initial phase. Okay, now it's insufficient. This is one parameter. One parameter, right, cannot code 2 coordinates. So let's understand this. So the important point is that a given torque that the animal can measure in its follicle gives it a locus of angles and a locus of radii. So each of those curves in the upper left-hand corner, each of those curves is a curve of constant torque. So if the animal senses the torque, all it knows is it's somewhere on that curve. So we need to measure, the animal has to measure a second parameter. So just as an example, how this could occur, okay, is that if the animal also independently, say, knew the initial phase of contact, it would be on one of these other lines. So say that's that black dot because it's a certain torque, and the black dot in the middle panel because it's a certain phase. These two curves cross at a point, boom. The animal has solved the computational problem. So this at least gave us a framework to think about going forward. And again, I mean, is this something that machine learning could help us with this thought process in planning our experiments rather than arguing on the blackboard? Okay, so now we're going to get into experimentation. So I described before, the object I called VERT is this oscillator. It drives the facial nucleus. This drives the vibrissa, drives the muscles, drives the vibrissa. And I want to look for how this coding occurs. And I want it to look at sort of the gateway to cortex, or the gates to cortex. So we're going to look at the output from cells in thalamus, VPM thalamus. But we're not going to do this by looking in thalamus. We're going to do this by looking at their projections into layer 4 of thalamus. And we're going to look at the presynaptic terminals. So over the last few years, Kasper Pogarski and his group has been building detectors of glutamate, the excitatory transmitter, okay, which would activate neurons in layer 4, both excitatory and inhibitory cells. But basically, this is a window to the world, okay, of thalamic output. And it has a lot of advantages because it stretches out the pulse a little bit in time. It makes, and this together with 2-photon adaptive optics developed in our lab lets us make progress. So the first thing we learn, these are just a couple of examples on top of torque from different single boutons. And I think the point is that torque basically is encoded logarithmically. So the fact that the model of the mechanics told you that torque decreases logarithmically as a function of length along the shaft begins to make some sense. This is basically Weber's law, okay? And in fact, if we look at this is the optical signal now from different boutons. And if we look at this as a function along the length, we find that it just falls off roughly linear, just like we found like the log of the torque falls off. And if you look at the peak torque as a function of contact phase, roughly it falls off like the sine of the contact phase. So the lesson is that vibrissatouch is encoded in terms of bending force, okay? But we need something additional. So the additional thing is to ask, is there a signal when the animal just whisks, independent of the touch per se, okay? If there's a signal just for free whisking in air. This is going back to the same experiment. Again, we're just labeling the boutons that we're recording in layer 4. And these are two examples, bouton 1, bouton 2. And that's the vibrissa angle that's recorded videographically. And we find out that different boutons preferentially give an extra spike, okay, depending on where they are in the whisk cycle. So maybe this will solve the problem, okay? And just to emphasize this a little bit, what you see is that, in fact, all potential phases are coded by this extra spiking, okay, of the boutons. So some phases have a higher representation than other phases, but all phases are covered. Okay, so what this means is that we have boutons that respond only to touch, or encode torque, and we also have boutons that respond both to touch and phase in the whisk cycle. So they're encoding torque and phase. And this is just, sorry for the brevity, these are just examples. The signal to noise is really quite good. And again, a lot of the credit goes to Kasper. So the bottom line is there's an additional finding that the neurons that spike in the absence of contact, okay, preferentially have their maximum signal at the same place they produce an extra spike. So about 1/3 of the units, okay, have this extra spike just when they're whisking without contact, and that's the most likely place for a unit to spike upon contact. And I think you can see this here. This is the relative phase between contact phi IC minus its preferred phase, and it's, you know, the peak of this is coming in at around 0. Okay, so let's put it together. So the lesson here is that thalamic boutons encode 2 signals that are a function of this polar coordinate, where you are along the whisker and your phase in the whisk cycle. And you know, whether it was half and half, it turns out about 2/3 are just purely coding torque, and about 1/3 are coding torque, plus this extra signal. I don't think the details matter. Okay, this is the data. So now again, how do we do this? Do we turn to a transformer or ChatGPT, whatever it is, 99? Or do we again go to the blackboard and argue? And this is a serious question. So this is the result from the blackboard, not from machine learning. The result from the blackboard is that there are cells that only encode torque. And this leads to a series of curves as a function of distance along the vibrissa and phase in the whisk cycle, where if you measure just torque, I know that one of these combinations of coordinates could be responsible for that torque. There are other units that code torque plus phase in the whisk cycle, get a set of different curves, and these curves will cross. Okay, and this is our running hypothesis for how the animal knows where it touched an object, okay, by having two classes of responders. So the converging story is that these thalamic inputs supply enough information for the animal to know where it touched. Now, I've left out a lot of stuff. I mean, there are other possible signals which we've measured, and they turn out to be very small. But cortex encodes the location of touch based on the confluence of inputs. One encodes only torque, one encodes torque plus a signal of self-motion. So a general contribution is we often think of self-motion as something to null out, right? You turn your head, and then your eyes rotate to counteract the motion of the head, right, in the visual ocular response. But here, we're using self-motion actually to give you a second signal that allows you to determine coordinates for exoreception. So at the end of the day, this brings up another point about this general challenge is how should data be uploaded? Do you give all the raw data, the many terabytes of raw data, which you may or may not have correctly sorted for spikes and calcium signals and glutamate signals, or do you give the cooked data? Do you give all the data that you took in the course of doing this experiment, including data that at the end you didn't publish or you judged to be auxiliary? Or do you publish all of it because maybe there's something you missed that somebody reevaluating your conclusions, your hypothesis could use, okay? And I mean, we've heard this morning about annotation, but I think this is very critical to know exactly what decisions were made in guiding your experiments. So if part of the reason to use open data is so that others can go back, have faith or not have faith in your raw data, and perhaps reinterpret it. Okay, thank you.
All right, thank you, David.
Just a comment on the last thing. I mean, I think, right, we probably would all agree that we want the raw data, we want the process data, we want the code that converts the raw data into the process data into models, and models into figures. That should all be there, right? That's really where we need to go.
I agree.
Yeah.
There might be a practical issue. As you know, if you have a couple of petabytes, But maybe that's just a transitory.
That's a
Yeah.
that's a separate.
I think
issue.
we
Yeah,
agree. I think that's very important. Yeah.
I thought the description of the phase preference, the evenly distributed phase preference of the thalamocortical boutons, to be curious. Forgive me if my memory is a little rusty, but I seem to recall that the activity in the follicle and in the trigeminal is driven mostly by the high velocity portions of the whisk. So how do you get from that to an even distribution in the thalamus?
So no, not to argue with your memory. It's not quite flat. Okay, it's not quite flat. But I think if you look, Frank Rice and more recently Mikkel Brecht have published on the anatomy of the follicle. So you basically get contact with this ring pulse at all parts of the motion. And the other thing to be a little careful about is, I mean, there are rapidly adapting receptors that you excite them once, you get a spike, and that's it. It's almost digital logic. But there are slowly adapting receptors that, so it's, I think this, I think the only difference between what we see here and what you see in the trigeminus, and I'm thinking more of Dan O'Connor's data, which I'm most intimate with, is that you see the effect of rapidly adapting receptors in the trigeminal ganglion, but we don't see any signatures where you get a spike at the beginning of touch and a spike at the end of the touch along this thalamic pathway. Okay, I mean, keep in mind that spinal trigeminal nuclei also have ascending and have this sort of rapidly adapting response. Okay, anyways, it's not quite flat, but I don't think this also makes perfect sense in terms of where you most likely touch. Yeah.
Max, did you have a question?
Thanks for a really lovely talk. I was wondering if you think the animal's whisker might be sensitive to the rate of change of torque, like the angular momentum, because it's going to get lots and lots of signals over time, and whether that might be useful information.
Yeah. Okay, it's a great question because I can answer it. Dan O'Connor made a lot of Kai Silverson in Dan's lab made a lot of intracellular measurements. I mean, this is rough, right? Because the trigeminal ganglia is sitting in a little cave, Beckles cave. So they report that there is a strong component of the d-angular momentum dt, okay, or d-torque dt. I'm sorry, d-torque dt. And we looked very hard. I didn't show it here because I had to keep it tight, but the change in torque with respect to time is flat, so it's not a good coding variable along this pathway. Okay.
Well, thank you.
I'm going to take the debate a little bit on your challenge about machine learning methods versus the Blackboard. I think you kind of made your view pretty clear on that. Do you see, and that's cool. Like challenge talks. That's kind of the point, I guess. Do you see any domain in neuroscience where you view machine learning models are going to be particularly well adapted in a way that Blackboard type approaches are not? I mean, one comment I can make is that you're modeling physical systems, which undoubtedly we've been the best at kind of blackboarding our way through physical systems in the way that physics has been very successful. I think for other problems, it clearly has not been so easy.
Okay, so look, we, like many people, we use deep lab cut, which is a statement that our simple ideas about looking for features of motion are not as good as what you're getting, using reinforcement learning on trained data, training data. So you got me, okay? But I mean, this is really as a means of analysis. We're looking at correlations. But I want to shoot this back at you. Physics, okay, has examples where great simplicities were discovered in what are enormous data sets. I'm not going to, just talk about throwing baseballs, but look at all the material data sets, okay, where we know about the crystal structure, we know all kinds of stuff about electronic states, band gap, et cetera, and try to predict some of the things you get from theories of superconductivity. So I kind of find it funny that in neuroscience, we're taking somewhat incomplete data and applying machine learning when you could take a much more complete data set, either from material science or chemistry, and see if you could discover those rules. All right, I would do that first before trying to work on a complicated data set like the brain.
A really nice paper recently where they took, a group in Oxford took about 30 years of planetary motion data and rediscovered Newton's laws of gravitation from a machine learning angle, which I think kind of maybe is kind of what you're talking about a little bit, like trying to rederive first principles from a machine learning perspective. I'm not sure if that was your point
No,
exactly.
that's the flavor.
Yeah, so I mean, people are working in that space, and I think it's, you know, interesting space to exist because it brings some of these kind of perhaps what you could view as you know, opposing views together to a similar common space where they could really help each other.
Yeah, I have maybe a different challenge, and I've taken the blackboard approach on this problem. But see, I have a totally different model in mind than yours, right? I don't think the phase has anything to do with it. We can discuss this. It's probably not of interest to this audience, but I think there are actually many different kinds of clues to get you radial and angular distances. A machine learning model, you could look, throw them all in there, and then ask, right, what would a machine learning model, for example, choose, right, in different things, and then you might get ideas, right, about how that might work neurally, and then you can. So, I think here you're taking a very clean approach, but it's really chasing one hypothesis, and you're seeing kind of a neural correlate in the data. You don't have behavior that these particular things, the behavior is the arbiter of truth ultimately, right? You have to do a behavioral manipulation to show that phase and torque is the thing that matters. You just show that they're encoded. There's many other things encoded, including axial force and all this kind of stuff that give you the clues. So I think actually a machine learning model could give you really an unexpected way of solving the problem, and then it's up to us to look for kind of, you know, through behavioral manipulation to see if that's actually what the animal uses.
No argument, right? There is one thing, let me just add to this. I mean, you could go back to information theory, which I think is important, right? In trying to understand which parameters, right, neuronal parameters, give you the most information, say, about the behavior. Now, information theory tells you nothing about the algorithm, right? But it does give you an upper bound. It does give you an upper bound, even a way to evaluate models. So I'm just throwing that into the mix. That should sort of be on the top, okay? And then there's no argument. Any means, machine learning, grandma has an insight. At the end of the day, you test it with behavior. No argument, yeah.